This video will help debrief the LeChatelier's principle experiment that we did in lab yesterday and help you answer some of the post-lab questions. You recall we did two different equilibria. Part A was the acid-base equilibria, which I'll talk about first. Remember that in that equilibrium we had an indicator which was called bromethymol blue, and it's symbolized in this lab as HIN, meaning that the indicator itself is an acid. It has a proton in the front. That's what the H is for in HIN. And therefore, if we mess around with pH, in other words, the number of H plus ions in solution, shown right there, then we can shift the equilibrium to the right or to the left. For example, if we add acid, then we're increasing H plus, and according to LeChatelier's principle, the equilibrium will shift away from the added component and produce more protonated indicator, which is yellow. So the opposite is also true. Let me just erase this. If we decrease H plus, if we do something to decrease the amount of H plus in solution, the system will shift to the right and create more H plus, and the byproduct is the indicator minus, or the IN minus ion, which is blue in solution. So the first part of that step was relatively simple. We added HCl. So if we add HCl, we are increasing this ion in solution. It will shift somehow to accommodate for that increase, and of course we saw that it turned yellow. The second part was a little trickier. It involved a second equilibrium. Remember, this is called the ion product constant for water, the dissociation of H plus and OH minus ions. What we did in this second step was we added NaOH, a base, right here. Now, according to the second equilibrium, when we add OH minus, this system should shift to the right and try to consume OH minus. But what's important for the first equilibrium, for the indicator, is that if this shifts to the right, right here, H plus is also consumed, so that will decrease. And if H plus decreases, now go up to the top equilibrium, if H plus decreases, so shown here, we're going to decrease this, the equilibrium shifts to the right, and you turn a blue color, and that's what we observed in the lab yesterday. Now, the second equilibrium that we messed with yesterday was the reaction equilibrium, which involved a couple different ions, the iron 3 ion and the thiocyanate ion, SCN minus. And we did a couple things to mess with this equilibrium. At first we put just a solution of iron 3 nitrate and we added some KSCN, or vice versa. I think we started with the KSCN. And it became a yellowish color. And then we can change the color to a dark red-brown by increasing one of these ions or decreasing them. Basically mess around with concentrations. Let me erase this. So there was a second equilibrium that came into play here, and that is the acid-base equilibrium. If we increase the concentration of OH minus in this system, in other words, mess around with pH, then that will affect the iron 3 concentration. And if you change the iron 3 concentration, either by increasing it or decreasing it, doing something, it's going to mess with this first equilibrium on top. And then, in fact, there is a third equilibrium that comes into play in this, because iron will change. Let me grab a pen here. Because iron will change if you mess with pH, then the pH equilibrium, again the ion product constant of water, will also come into play. So here's some things that we did in this lab. First is that we added iron 3 nitrate, and I think that's obvious. The effect here, if we add iron, the equilibrium will do something and shift to compensate for that change. So I think that's relatively obvious. The other thing we also did, which should be obvious, is we added KSCN, potassium thiocyanate. And also, in this case, it should be obvious. We're increasing this ion in solution, so the equilibrium, again, will shift. The third one is a little tricky. We added potassium nitrate to solution, and most of you, if not all of you, did not observe any change. Is that surprising based on the equilibria that concern us? If we dissolve this stuff in water, we get potassium ion K plus, and we get the nitrate ion NO3 minus. And notice, do those species take part in any of the equilibria that we're concerned with? No, they do not. So they do not have any effect on the equilibrium. In fact, notice that iron 3 nitrate has nitrate, potassium thiocyanate has potassium in it, but they are not the reactive species that change color in solution. All right, let me erase all these markups here. There we go. Now the next thing that we did to this equilibrium on top here is we added a species that's called sodium phosphate monobasic. The actual, I think the better name for it is sodium dihydrogen phosphate, but this idea that it's called monobasic down here is actually really important for the chemistry of our equilibria. You may recall that when we added this stuff, this solid, we saw a pretty cool color change. Here's a quick movie of what we saw. We added the solid sodium dihydrogen phosphate or sodium phosphate monobasic to the system shown here, if you can see my arrow, to the system that had a lot of FeSCN2+, meaning that blood red substance in solution. And when we added the sodium phosphate monobasic, we saw this occur and swirled. That blood red material was consumed, and we saw a pretty cool color change where all of that blood red iron thiocyanate ion was consumed and disappeared completely. Thanks to Chris Fleischer for being an awesome hand model, by the way. So let's go back to the notes and try to figure out what's happening here. It's called monobasic for a reason. Let me go to the next slide and show you what's going on here. Sodium phosphate monobasic is a Bronsted-Lowry base. Now, harken back a few months to remember, what is a Bronsted-Lowry base? A Bronsted-Lowry base is something that absorbs H+. Remember, the definition of an acid doesn't change, something that donates H+ to solution. But a Bronsted-Lowry base absorbs H+ from solution. So let's take this substance, the sodium phosphate monobasic, and let's dissolve it in water. So that's shown here. If you take the sodium phosphate monobasic and dissolve it in water, you get sodium ion. That's always soluble. Remember SNAP. And you get this ion in solution, which is the dihydrogen phosphate ion. This is a Bronsted-Lowry base, and it does this when it's surrounded by water, which of course it is. Look, we go from H2PO4 minus to H3PO4 in solution. And in the process, and here's the cool part, in the process, it splits water. It absorbs that hydrogen from water, and what does it leave behind? OH minus in solution, and therefore it acts as a base. So we go back to our three equilibria here, and look what we've done. When we dissolve the sodium dihydrogen phosphate in water, we increased in water this species right here, OH minus. So down to equilibria A2. We've increased this in solution. What does that bottom, well, we've increased OH minus in solution. Actually, we could even use this one right here. If we increase OH minus in solution, this equilibrium is going to shift to the right. When it does so, it first of all makes it precipitate. Some of you saw that. But second of all, it consumes iron. Now that's really important for this top equilibrium right here. If it consumes iron, that means that this species in the very top equilibrium is decreasing. What is this top equilibrium going to do to shift which direction? To compensate for that decrease in iron, it's going to shift to the left, away from the colored species, the red-brown species, which is consumed. So here is a case where we have three different competing equilibria, three things going on because of a change in pH instituted by a Bronsted-Lowry base, a material that absorbs a hydrogen ion from solution, but again, importantly, leaves behind the OH minus. And wow, that's confusing, but also kind of cool. Good luck, and I will see you later.